Before we dive into the intricate process of RNA isolation, let's take a moment to appreciate the brilliant minds that made this all possible. The discovery and development of RNA isolation was a collective effort of numerous scientists dedicated to understanding the very fabric of life. Among these pioneers, we remember the significant contributions of James Darnell and Howard Temin, who in the 1960s laid the groundwork for understanding RNA and its role in gene expression. Their work paved the way for the development of techniques to isolate RNA, a critical step in studying gene function and cellular responses. But the journey to this discovery was not easy. It was a winding path filled with challenges and setbacks. There were numerous failed experiments, countless hours spent in the lab, and many hypotheses put to the test. But with every failure came a new opportunity to learn, to refine, and to persevere. The breakthrough finally came with the development of effective and efficient protocols for RNA isolation. This discovery has since powered countless research studies, enabling us to understand diseases, develop treatments, and even create vaccines. Now that we've acknowledged the pioneers of RNA isolation, it's time to delve into the process itself. So buckle up and get ready for a microscopic journey into the heart of molecular biology. Welcome to the fascinating world of RNA isolation. In this crucial molecular biology procedure, we get to explore gene expression, identify diseases, and even develop vaccines. Essentially, RNA isolation is the extraction of RNA from a biological sample, which involves a series of calculated steps. The starting volumes for the process are typically 1 million cells, 1 milliliter of trizol, and 10 UL of beta-mercaptoethanol. Once we've got the right quantities, the first step is to disrupt the cells to release the RNA. Then the RNA is separated from the other cellular components. Lastly, it's purified and kept for future use. It might seem straightforward, but each step is a world in itself, requiring accuracy, comprehension, and the right chemicals. Speaking of chemicals, it's key to highlight the essential safety precautions we must take. Due to the hazardous nature of some of these chemicals, it's vital to wear protective lab coats, gloves, and eye protection to avoid any potential contact with skin or eyes. We must always remember that laboratory equipment such as pipettes, centrifuges, and microtubes must be handled with care to ensure a smooth process and safeguard against contamination. Now let's bring attention to our key players: trizol with added beta-mercaptoethanol, chloroform, and isopropanol. Trizol, along with beta-mercaptoethanol, is our cell disruptor. Chloroform aids in RNA separation, and isopropanol is used for RNA precipitation. Each chemical is instrumental, and we'll delve into their specifics in the following scenes. Are you ready deep into the intriguing world of RNA isolation? Ensure your lab coat's buttoned up, your gloves are on tight, and your goggles are secure because we're about to embark on a microscopic journey. Welcome to Scene 3. Now let's meet trizol, the star player in RNA isolation, and its powerful sidekick, beta-mercaptoethanol. These are not just chemical compounds, but precision tools in the world of RNA isolation. Beta-mercaptoethanol plays a key role in enhancing trizol's work efficiency. So what exactly do trizol and beta-mercaptoethanol do? They act as unique keys, unlocking the cellular vault to uncover the hidden treasure within, the RNA. Consider RNA isolation as a scientific heist, and trizol, complemented by beta-mercaptoethanol, is our master key. But instead of opening doors, these chemicals dismantle cells, acting on the cellular and nuclear membranes. The active ingredients in trizol are a monophasic solution of phenyl and guanidine isothiocyanate, GITECC. Phenyl, encoded by the chemical formula C6H6OH by denaturing proteins and disrupting their structures, while beta-mercaptoethanol assists by further breaking down disulfide bonds in proteins. Guanidine isothiocyanate, featuring a chemical formula of C2H6N4S, inhibits RNase activity, guarding the RNA from degradation. When trizol and beta-mercaptoethanol are added to a sample, cell lysis occurs, a process where the cell membrane is releasing the cell contents into a solution. Looking at trizol's part in the phase separation step, it dissolves lipids and proteins, ingredients of the cellular and nuclear membranes, resulting in the liquefaction of the cell. This process produces a solution containing RNA, DNA, and proteins. The breakdown of lipids and proteins forms a biphasic mixture. The upper aqueous phase, which houses the RNA, can then be meticulously extracted. In the end, RNA is precipitated from the aqueous phase by adding isopropanol. The interaction between isopropanol's hydroxyl group and the RNA's phosphate groups leads to RNA precipitation. This crucial step, further improved by the presence of beta-mercaptoethanol, allows for effective RNA isolation from the rest of the cell components. Trizol's unique ability to selectively isolate amplified by beta-mercaptoethanol is due to specific chemical interactions. It interacts with other compounds to break down protein structures that could otherwise degrade the RNA. Basically, trizol and beta-mercaptoethanol serve as protectors, shielding the RNA from any potential degradation while the isolation process is ongoing. This sophisticated and eloquent approach to RNA isolation is why trizol, heightened by beta-mercaptoethanol, is a crucial part of the equation. Therefore, trizol beta-mercaptoethanol are our keys to unlocking cellular secrets and obtaining the invaluable RNA. Scene 4. Trizol can't do it all alone. Let's introduce chloroform and isopropanol, the two essential components in RNA isolation. Let's dissect their roles and the specific quantities we require for each stage. Initially, we incorporate 200 microliters of chloroform into our specimen. This instigates a division, enabling us to recognize the individual layers that surface. We then vigorously agitate this concoction for 15 seconds, merging the trizol and chloroform into a single uniform solution. Allowing it to rest for 2 to 3 minutes, the concoction segregates into 3 phases. The uppermost aqueous phase, which contains the RNA, is our focal point. Chloroform's function here cannot be undermined as it ensures this separation. Once we've segregated this layer, we're set for the subsequent step, the advent of isopropanol. We use 500 microliters of isopropanol to precipitate or solidify the RNA within the aqueous phase. After stirring, the RNA precipitates, a process we commonly expedite by keeping the concoction at -20 degrees Celsius overnight. The next day, we utilize centrifugation to segregate the RNA, which has now formed a pellet at the bottom of the tube. Then, we cautiously eliminate the supernatant, leaving behind our RNA pellet. A swift rinse with 1 milliliter of 75% ethanol eradicates any lingering impurities, yielding our purified RNA. As you can see, the RNA isolation process is a detailed dance of specific quantities and constituents. Chloroform and isopropanol are our critical allies, helping us in the separation and precipitation of the RNA. With precise quantities of chloroform and isopropanol, we've captured our RNA precisely where we wanted it. Having isolated the RNA, our journey continues. Now that we've separated the RNA from the rest of the cellular components, we move on to the purification process. This stage is crucial to ensure that the RNA is free of any contaminants that might interfere with downstream processes, such as DNA or proteins. This is typically achieved through a series of wash and spin procedures using ethanol or similar solvents, which help to clean the RNA without degrading it. You might wonder why we're so cautious about RNA degradation. Well, RNA is a rather delicate molecule. It's prone to breakdown if not treated with care, and this is why we use RNase-free reagents and equipment throughout the process. These precautions help to maintain the integrity of the RNA, ensuring that our sample is of high quality and suitable for further analysis. Once our RNA is purified, we need to store it properly to prevent any degradation over time. The optimal storage conditions for RNA are quite chilly. We typically store RNA at -80 degrees Celsius. Yes, that's colder than your average winter day in Siberia. This extreme cold helps to slow down any enzymatic reactions that could degrade the RNA. It's also important to minimize freeze-thaw cycles, as these can also lead to degradation. So when you're retrieving your RNA from storage, take out only what you need and promptly return the rest to the freezer. But what if you need to use the RNA right away? Well, you can store it in RNase-free water or buffer at 4 degrees Celsius for a short period. And remember, always handle your RNA with care. Use gloves and clean equipment to avoid introducing any RNases that might be lurking on your skin or in the environment. And voila, we've got our RNA, pure and ready for storage. As we step into scene 5 of our journey through the RNA isolation process, rest assured knowing that your precious RNA is safe and sound, ready for whatever experiments lie ahead. Scene 6. What's next after our RNA is ready? Let's find out. A plethora of successive steps after RNA extraction are found within the broad domain of molecular biology. Nevertheless, before we delve into these procedures, it's imperative we probe into the different subspecies of RNA. Each subspecies has a distinctive function and relevance in the biological system. We begin with messenger RNA, commonly known as mRNA. This RNA variant is the courier of genetic information, ferrying DNA from the nucleus to the ribosome, where proteins are synthesized. Next, we have ribosomal RNA, or rRNA, which constitutes a crucial part of ribosomes. Then comes transfer RNA, or tRNA, that aligns amino acids as per the instructions of mRNA, aiding protein synthesis. Small nuclear RNAs, or snRNA, another member of our RNA family, play an essential role in splicing and other RNA processing events. We also have microRNAs and long non-coding RNAs. The former modulates gene expression while the latter carries out diverse roles in gene regulation. As we go through each type, 3D animations and imagery representing these RNA types will be displayed on your screen. Now that we've acquainted ourselves with the various RNA types, we can move on to the first step in most procedures, reverse transcription. Here, the isolated RNA is transformed back DNA by an enzyme aptly named reverse transcriptase. This chemical reaction gives birth to DNA-RNA hybrid molecules. Subsequently, the RNA strand is degraded and replaced by DNA, resulting in a complementary DNA or cDNA. This cDNA mirrors the original RNA, acting as a standby for the original sample. Progressing further, we come across another pivotal procedure, quantitative PCR or qPCR. In this method, the RNA turned into cDNA is used to ascertain the expression levels of specific genes. The process involves a cycle of heating and cooling that prompts the DNA to unwind and replicate. This can be compared to taking a snapshot of the genetic activity at a certain point, furnishing invaluable insights into biological functions. However, each procedure has its own set of difficulties. Contamination is a frequent problem, possibly arising from proteins, DNA, or enzymes capable of degrading RNA. The solution lies in caution and patience. Always use RNase-free reagents, ensure your gloves are fresh, and sterilize your workspace before and after every experiment. A low yield is another common problem, usually caused by RNA degradation during isolation. To prevent this, keep your samples at cooler temperatures, limit their exposure to air, and shorten the time between isolation and subsequent processing. In case of inconsistent results, the culprit could be variations in the quantity of your starting material or disparities in RNA quality. Always start with a consistent amount of material and ensure your RNA is of superior quality. A spectrophotometer can be used to verify the purity and concentration of your RNA. Remember, results validation is paramount. Always incorporate positive and negative controls in your experiments. These controls serve as a guide, helping you determine the accuracy of your procedure. Now, equipped with a thorough understanding of RNA, its types, and practical advice and tactics, you're well on your way to mastering the fundamentals of RNA isolation. Scene 7. We've come a long way in our RNA isolation journey, haven't we? We've dissected the crucial roles of triazole, chloroform, and isopropanol. Our reagent disintegrates cell structures and grants us access to the RNA within. Chloroform then steps in, separating the RNA from cell components and creating a clear, RNA-rich supernatant. Isopropanol follows suit, precipitating the RNA out of the solution and giving us a tangible, workable product. We've also explored the rigorous purification process to rid our RNA of any unwanted contaminants. We've emphasized the importance of optimal storage conditions in preventing RNA degradation and delved into the consequent processes, including the use of isolated RNA in further experiments. Possible obstacles and their appropriate countermeasures were also discussed. Each chemical and each step carry specific volumetric units and require careful handling to maintain the quality of the RNA. And there you have it, a summarized overview of the intricate process of RNA isolation, with its chemical reactions, volumetric units, and safety measures. Until we meet again, continue your exploration of the microscopic world. Before we conclude Scene 8, let's highlight some additional safety reminders and ethical considerations. While we've shared this information to provide a comprehensive understanding of the RNA isolation process, It's important to remember that the information shown in this video is solely for educational purposes. Attempting to replicate these procedures outside of a controlled laboratory environment can be dangerous and is strongly discouraged. Furthermore, any experiment involving RNA isolation should be performed under the guidance of a knowledgeable supervisor and with the necessary safety equipment in place. Moreover, the scientific community is built on a foundation of responsibility and ethical behavior. Therefore, before performing any such experiment, an in-depth review of the relevant literature is crucial. Approval from the appropriate authorities, such as institutional review boards or ethics committees, is a must. These steps are not only important for your safety, but also for ensuring the integrity of your research and the scientific community as a whole. So as you continue exploring the microscopic world, remember to always prioritize safety and adhere to the ethical guidelines set by the scientific community. Happy researching!